Okay, so today I'm going to be presenting a little summary of mosquitoes and coronaviruses and how they might interact with each other. So, an outline, I will be dissecting some commentary that was done by George Poinar Jr., and he's from the Department of Integrative Biology in Oregon State University. And he has written this commentary regarding the potential role of mosquitoes in transmitting coronaviruses, or specifically the SARS coronavirus 2 that's causing the current pandemic. So it's going to start with an introduction of the commentary, and then we're going to move on to the criteria of becoming effective vectors. And we are then going to look at which criteria have already been met by this particular coronavirus. And then we will end it with future topics because I'm not just going to do this video. I'm going to continue with it, going a little bit further into depth later on. So, introduction, and this is the particular paper that I'm going over. It's Could Arthropod Vectors Play a Role in the Spread of COVID-19? And I will have a link for this particular commentary in the description below. So, currently, the novel COVID-19, a single-stranded RNA spherical virus in the family Coronaviridae, infects, like viral pneumonia and influenza, respiratory passages and spreads mainly by airborne infectious agents. So these are respiratory droplets from coughs and sneezes. Virus particles, or virions, also occur in the blood, intestines, and feces. So keep that in mind, that it is also present in the blood, intestines, and feces of human subjects. While at present, there is no direct evidence that COVID-19 or any of the SARS viruses are arboviruses. So those arboviruses means arthropod-borne viruses. So that means mosquitoes or other types of insects that might be able to get into contact with you and get into contact with something infectious and then can transfer that to a mammalian or some other animal host. So, where were we? What are the chances that the arthropod vectors could play a role in the spread of or severity of COVID-19? So, that was the introduction. Just one comment is that COVID-19 is a single-stranded RNA spherical virus. So that means that it's an RNA virus, which so its genetic material uses RNA rather than DNA, which is in your own cells. And so that means that when this virus does end up infecting your cells, your own machinery is going to turn, or I should say, read the RNA and use that as codes to create proteins for the virus itself. Now, the first protein that it makes is what is known as the RNA-dependent RNA polymerase, which is basically the copying machine of this RNA. So what it's going to do is it's going to make the initial RNA-dependent RNA polymerase. That RNA-dependent RNA polymerase is then going to read its own viral material and duplicate it and make many, many strands. And in that entire process, the virus is able to make a bunch of different other RNA sequences that can be used for making new proteins, and then those new proteins are then read by the machinery of your own cell to produce a virus particle. So keep that in mind. Your body, your own cell, is completing the job of the viral DNA. And a lot of times this is specific for whatever creature the virus is in. So, I read this already. So down here, while this scenario could theoretically be possible, there are several factors that would have to be overcome before it could become a reality. Due to their cosmopolitan distribution, wide host range, ability to transmit a number of different viruses from wild animals to humans, and from human to human, many hematophagous arthropods, so hematophagous meaning like hema, blood, phagus, eating, so blood-eating arthropods, insects, And these are mosquitoes, biting midges, black flies, sandflies, and ticks are all available for vectoring COVID-19, with mosquitoes being prime candidates. So basically, we're not saying that it is happening, but that if it is happening, mosquitoes are probably going to be a prime candidate. So before a virus can be transmitted by a blood-sucking arthropod, it must meet two criteria. The first, the first criteria is that the virus must occur at least during some period during the infection in the blood of the vertebrate host at a high enough titer or concentration to be acquired by the arthropod. So that means, so a lot of viruses, you catch them, they do not end up going into the blood. A lot of them, like some colds, for instance, it goes into your lungs, it goes into your respiratory tract, and it stays in that area. And if it stays in that area and is only multiplying in the cells of your lungs, then it might not get into the blood in a high enough concentration for it to be bloodborne. But if that does happen, it is known as viremia, so viremia in the blood. You can get that happening, and if that happens, then a mosquito or some other insect might be able to bite you, and they might be able to get a sufficiently large dosage of the virus when they do that bite. And this doesn't happen with many diseases, but that is one of the prerequisites of something being able to be transferred by mosquitoes. So that's the first criteria. And second, for biological transmission, the virus must pass through the gut wall of the vector, infect and replicate it in its salivary glands to reach a high enough titer to ensure transmission to the vertebrate host. So once the virus ends up getting inside of the mosquito or whatever arthropod host it ends up getting into, it must be able to multiply within that particular host. Now, remember when I was talking about how the virus gets into your mammalian cell or your lung cells and it uses the machinery, the biological machinery, to duplicate itself? This might not necessarily be compatible with machinery that is in a mosquito, or it might not be compatible with machinery that is inside of black flies. So for the virus to be able to become infectious within the mosquito cells have to be able to take up the virus and then multiply within the mosquito as well. And this is pretty rare. This doesn't always happen. And that's because for a virus to be able to go between two very genetically distinct creatures, it needs to have evolved or come across some way to have take over the machinery of two completely different things. So a lot of diseases can easily cross between humans and monkeys because our immune systems and our biology are so similar that it doesn't take a lot of mutations to do that cross. So for instance, Ebola was very easy to be able to cross between humans and primates, or things like HIV. There's simian HIV, which doesn't do as much damage in simians, but ends up causing a lot more damage in humans. So viruses can easily make the jump between very similar creatures, but that is not necessarily the case where if you might, it would be very difficult for a virus to end up infecting both an insect and a human simultaneously. It is possible, but if it does happen, it needs to employ strategies that are universal or ubiquitous between mammals and arthropods. And I shouldn't say just mammals, I should say vertebrates. So, and that's the first option. The second option is just mechanical transmission. So, I can't read it right now, but mechanical transmission could occur if an infective dose of the virus is on the mouth parts of the blood-sucking arthropod. So this means if let's say the virus is not able to multiply within the host a mosquito or some other insect that bites you. If that insect can then quickly, very quickly, bite you and then bite someone else within a few hours. I mean, I'm saying a few hours, but who knows how long the virus can last within the mouth parts. If it can do that, then it is possible that it ends up transferring from the mouth parts of the mosquito or the other insect into its next victim. Um, that's more difficult to happen because since the virus is not multiplying greatly, it's not going to have a large viral load within the mouthpiece of the creature, but it is still possible. So, and here's a photo of a potential vector of COVID-19. This is Culex tarsalis, which is now a primary vector of West Nile virus, Western equine encephalitis virus, and St. Louis encephalitis virus, all of which infect humans. So this is possible. It is possible for viruses to infect both the mosquito and the human, but this is not known to happen with SARS-CoV-2. So studies show that COVID-19 may spread in several ways in infected humans, including via the nose or mouth, intestines, feces, and blood. In discovering a blood filter used to treat COVID-19 patients, a new release stated that very recent publications and clinical feedback confirmed the presence of the virus RNA in the blood of critically ill COVID-19 patients. So, going back to the first criteria, we now know with pretty much certainty that at least in severe cases of COVID-19, that the blood does contain detectable levels of the RNA of the virus. And usually that would indicate that if you can detect the RNA, there's probably most likely very viable viruses that are floating around in the blood in concentrations that are high enough to detect. So, and since we know how infectious this is, if you can detect it, it probably means you're going to get infected, or it's infectious. So that's one criteria down. And which criteria maybe? Okay, so coronaviruses infect a wide range of animals, and in some cases, the titer of virus in blood would appear to be high enough to be acquired by biting arthropods. There are two feline coronaviruses that infect cats worldwide. In the feline enteric coronavirus, the virus is limited to the intestines and the virus particles are shed with the feces. Biting arthropods probably would not be able to acquire any virus particles in the blood of infected cats. So this, yeah, okay. However, with the feline infectious, I don't know how to pronounce this, but peritonitis coronavirus, which is antigenically related to and serologically cross-reacts with a subgroup of mammalian coronaviruses. So let me stop right there. Antigenically related to and serologically cross-reacts means that the virus is presenting proteins on its surface. And those proteins of this feline infectious peritonitis coronavirus are similar enough to the mammalian versions of coronaviruses, or I should say some of the mammalian coronaviruses, that if you have immunity, if you have produced antibodies for one of them, they're very likely going to interact with the other. So that could confer some immunity. That could confer some immunity to you if you end up catching one and then you end up catching a different one later. So, including the transmissible gastroenteritis of swine, human coronavirus 229E, and canine coronavirus, cats can develop a persistent viremia. So keep that in mind. Cats can develop a persistence viremia, so they can have virus in the blood persistently. And this is just one example of a feline coronavirus that is able to do that. So blood-sucking arthropods would have a good chance of obtaining the FIPC virus when they feed on infected cats. So if the titer of COVID-19 in the blood is high enough, the first criterion would be met. The virus could be taken up by blood-sucking arthropod vectors. Still unknown is whether enough virus would replicate inside of hematophagous arthropods to initiate a biological cycle. If some type of receptor cells occur in the arthropod vector, a biological cycle could be initiated. Otherwise, the transmission would be mechanical, with the virus particles carried in or on the mouth parts of the potential vector. Mosquitoes are very successful in transmitting single-stranded RNA spherical viruses of the families Flaviviridae, Togaviridae, and Buniaviridae to humans. While these viruses cause fevers, body aches, encephalitis, nausea, vomiting, and hepatitis, they typically do not develop in the lungs of their victims, as does COVID-19. However, some of the reservoir hosts of these arboviruses are the same as those of COVID-19. So, going back to that, at least of the single-stranded spherical viruses, there are known viruses that can go between mosquitoes and humans. However, there is not any known right now of any coronaviruses. So let's keep that in mind. No coronaviruses are known to go between humans and mosquitoes. So, continuing, since both coronaviruses and flaviviruses are abundant in bats, including insectivorous horseshoe bats of the genus Rhinophis, which are thought to be possible to be reservoir hosts of COVID-19, it could theoretically be possible for mosquitoes to acquire virions of both COVID-19 and flaviviruses when feeding on infected bats. Whether these mosquitoes could transmit COVID-19 along with other flaviviruses to both bats and humans is unknown, and that is unknown as of yet, which is why a lot of research has been thrown into researching coronaviruses and other diseases of bats, because we simply do not know a lot of this information. So continuing, the original reservoir hosts of the deadly yellow fever arboviruses appear to be bats and rodents living in endemic lowland equatorial Africa and South and Central America. In its enzoonotic form, the virus occurs in monkeys and is spread by forest mosquitoes. In the epidemic form, it occurs in humans and is spread by mosquitoes of the genus Aedes. When obtaining the yellow fever virus from bats, the mosquitoes may also be acquiring coronaviruses. Equine encephalomyelitis is caused by arboviruses of Togaviridae, with birds as the primary vertebrate hosts and mosquitoes as the vectors. Venezuelan equine encephalomyelitis strain is especially interesting since horses infected with epizoonotic strains of VEE have a persistent and significant viremia, which could make it possible for the virus to be acquired by mosquitoes. But infections can also pass from horse to horse by respiratory secretions and direct contact. So here is a virus that has two possible methods of transmission, one by mosquitoes and the other by respiratory viruses. So remember, going back here, they're talking about all these viruses that are known to be transferred between humans and mosquitoes, but none of those were respiratory viruses. However, in an equine model, we do know that there is a disease, at least one disease, that is respiratory. It can go between horses just breathing on each other, or I should say, getting their droplets on each other. But it is also transferred by mosquitoes because the virus exists in large enough quantities in the blood of the horse, and it is also somehow able to multiply within mosquitoes. So what I'm going to discuss later is what are some of the strategies that a virus can employ to be successful in multiplying within the mosquito host and then also getting into the blood of the creatures that they infect, in the mammals that they infect or the vertebrae that they infect. Because there can be distinct strategies that have been researched on how a virus might be able to mutate into a more infectious disease. And that's going to be a follow-up video. So, interactions between vectors and viruses are mostly unknown since the dynamics are constantly changing. Since flaviviruses and togaviruses can replicate in both insects and vertebrate hosts, they have a high capacity for rapid evolution into strains of varying virulence and host selection. So this is what I was talking about earlier. You have your own machinery within your cells. The insects have vastly different machinery within their own cells. And so a virus that is able to go in between a mammal or a vertebrate with an invertebrate or an arthropod or an insect, it needs to be able to either evolve rapidly, which is very likely, or it's just very lucky. So mutations can result in a shift to new hosts as well as a change in developmental patterns in original hosts. This has been noted in the case of West Nile transmitted by Culex pippins. We still don't know what mutations are possible with COVID-19. So there is a lot of indication right now that this particular virus, COVID-19, SARS-CoV-2, multiply or evolves slowly, that it mutates slowly. So we can do phylogenetics, or we can look at the mutation rate of this particular virus, and we see as of now, as of yet, it mutates relatively slowly. That does not mean necessarily that the phenotype is evolving slowly. So sometimes you can have just a very few select mutations that are very good at evolving the creature or evolving the virus as a whole. So, so yeah, we still don't know what mutations are possible with COVID-19. However, there are a few mutations between COVID-19 and other coronaviruses that are pretty notable, and I will discuss in following videos. So, if tissues of the body other than the lungs and intestines become infected with COVID-19, the titer of the virus in the blood could increase, making more virins available for blood-sucking arthropods to acquire when feeding on infected humans. Blood from victims of COVID-19 that are homeless or in refugee camps could supply high levels of virus to hematophagous arthropods. So we really need to keep this in mind as well, is that if we do let this virus spread throughout the community in large quantities, we are then giving it a lot more opportunities for a mosquito to bite the people and then give the virus a chance to mutate into being able to infect mosquitoes as well. So the more opportunities we give it, the more opportunities it has to become mosquito-borne. So if we can suppress everything as much as we can and keep the viral loads low in the blood of people, then and get them off the streets, keep them from getting bitten by mosquitoes. If we can accomplish that, we can reduce the likelihood of this becoming mosquito-borne. And continuing, as far as known, there is no evidence that variants of COVID-19 or of any coronaviruses are carried by hematophagous arthropods. Without some very substantial mutations, it is unlikely that biological transmission of the virus by blood-sucking arthropods would occur. Now, let me stress here, I have seen some evidence that some very, very important mutations have taken place in the COVID-19 or SARS-CoV-2 version of coronaviruses that would make it more susceptible to being able to infect mosquitoes. And that some of it has to do with some very important mutations in cleavage sites of the spike protein, so one of the spike proteins that is coming off the virus. And I'll do quite a few videos on the subject later. So where am I? So successful transmission could theoretically be possible if the infected virus particles were physically carried on or in the mouthparts of potential vectors. However, that's much harder to accomplish because you would need a mosquito to bite one person and then bite another person within a relatively short period of time. But if it can multiply within the mosquito, then it has days to fulfill that function. So other pathogens, virus, bacteria, protozoa, etc., introduced into patients by hematophagous arthropods could well increase the severity or delay recovery from COVID-19. Yeah. So, and yeah, open access. So, which criteria met? So, for future topics, what are the mechanisms? I'm going to do a video on this. What are the mechanisms of how viruses can transfer between hosts? And one of them is a protease review, and another one will be how viruses actually are able to replicate in the cells. So that's going to be another future topic. And then another topic is what substantial mutations or recombinations aid in insect vector multiplication. Does SARS-CoV-2 have indicators or effective crossing? Um, and then, so yeah, first video, so things are a little rough, but subscribe to notifications to be alerted to the follow-up videos that I give later. So, and if you have any questions, please leave them down in the comments. And, I'm trying to rush through all these things, but, it takes a lot of time. So, thanks.